Hello, biologists. Today we're going to talk about Mendel and Punnett squares. Gregor Mendel was a German friar, and a friar's like a monk. He really wanted to be a scientist, but back in the day when he lived in the 1800s, the only access he really had to literature and scientific materials was through the church. And so he joined as a friar so that he could experiment with his plants. He loved plants. He lived from 1822 to 1884, and his thing was breeding pea plants. What's a pea plant? A pea plant is this beautiful plant right here. So it's what we get snow peas from or sweet peas from. Not the same thing as a bean plant, but peas. We also have like English peas that you would have in rice, you know. So here's a beautiful pea pod. Inside the pea pod, you have pea seeds. A seed is the next generation of pea plants. So peas reproduce like this. They produce these gorgeous flowers. Some are pink, some are purple, some are white. They're all sorts of different colors. I'm growing peas outside right now, and they're this beautiful, like, blush color. They're really pretty. These flowers are considered perfect by botanists, which means that they have both male and female parts. The male parts are called anthers, and they are up on these long tubes, and these anthers produce plant sperm, which we call pollen. Here we have the female parts. At the top, we have a stigma, and then this is the style, and then down here is the ovary. So the ovary is where you contain the eggs, and this is the ovary in its entirety. When these eggs are fertilized, they'll develop into seeds like we see right here. So every time you eat something with seeds, you're eating an ovary, a plant ovary. How this works is a bee or some other nice pollinator will come over to this flower. It will touch these pollen sacs called anthers, and bees buzz. The frequency that they buzz at helps jiggle the pollen out of the pollen sac, and it covers their little body. And then as they move around from flower to flower, they are transmitting pollen from flower to flower. So they'll take the flower from here and use it to fertilize the eggs here. And that's how you get these beautiful seed pods full of the next generation of plants. So we have two generations shown in this picture, the parent generation, which is the pea plant itself, and then the next generation, which is the seeds, which will grow into a new plant when they're planted. What did Mendel do? He bred pea plants, and he did it because they are small, they are easy, they grow fast. In fact, if you want to start a garden, I recommend starting with pea plants. They're delicious and they're pretty easy to grow. The way that you can control their breeding is simple as well. So all you have to do to control who pollinates who is remove the anthers. So Mendel snipped away the sperm from this flower, and he would take the sperm from the flower he wanted to fertilize the original flower, and then he would dust that sperm onto the female part. So he's adding the pollen to the stigma, and that would ensure that all of these eggs are fertilized by this second flower here and not accidentally fertilized by itself. Self-pollination is pretty common in plants, and so he had to get rid of those. That would be one of his confounding variables. And then what he did is he allowed those seeds to develop, and each of those seeds would grow into a new plant. And so he could see how the traits were passed down from generation to generation. Mendel's whole thing was doing this hundreds of times and keeping like a great journal of it. So he looked at 7 different traits. First thing he looked at was flower color. There was either purple or white. Plant height, tall or short. Seed color, seed shape, whether or not it was brown or wrinkly. Pod color, pod shape. Was it smooth or was it bumpy? And then flower position. Axial means armpit. So an axial flower grows in the armpit between a leaf and a stem, and terminal means top. So a terminal flower grows right on the top of the plant. So he did this hundreds and hundreds of times, looking at these 7 traits, and he found that some traits came up way more than others, and so he termed them dominant. The other traits that came up less were termed recessive. Now you have to remember that Mendel didn't know about genetics. He didn't know about DNA. He didn't know about genes. This was before we had that vocab. The idea back in the day was just that you took two things and you blended them together. And so everything based on the understanding he had should have been an intermediate, right? Like it should have been purple plus white flowers equals light purple flowers. But instead, he was getting purple plus white flowers equal purple flowers. And that was confusing. And then he would breed the second generation with itself, and he would get purple and white flowers. And he was super confused by it. And so he took these notes and he meticulously did it. And he studied these pea plants just hundreds and hundreds of times. We now can track what he was watching in something called a Punnett square. And so a Punnett square is a theoretical breeding between two organisms. So let's look up here. We're going to have some vocab we need to know. The first generation that you start with is the P generation. That's parental. That's easier to think of it as the parent generation. Mendel termed them true breeding, meaning that when he bred purple flowers with themselves, he always got purple flowers. When he bred white flowers with himself, he always got white flowers. So then he made a cross, and he took a purple flower and a white flower, and he ended up with all purple offspring in the first generation. So F1 stands for first filial generation. Filial means family. So we have the first filial generation. That means that these are the children of the P generation. He would then take the F1 generation and he would cross it with itself. So he would use pollen and eggs from a single flower, and he would end up with the F2 generation. In the F2 generation, he had about 75% purple flower and 25% white flower, and that was not what he expected. And that's part of his curiosity about these experiments, is he would just keep going and tracking and seeing what happened. The F2 generation is the second filial generation. These are the children of F1 and the grandchildren of the P. And then your F3 generation, if you continued breeding single flowers with itself, that's called a self-cross, he would get all purple, 75% purple, and 25% white, and then all white. And so he found these really interesting patterns that he didn't have the vocab to explain, but we do. And so we'll explain it with vocab. The third filial generation is the grandchildren of F1 and the great-grandchildren of F2. And this can continue. You can have F25, but it's just tracking what generation number they are. So P is parents, and then second generation, third generation, fourth generation from the parents. Here's Mendel's notes. He identified the dominant trait, the recessive trait, and he kept breeding these plants. That ratio is almost 3-to-1. He did the same thing for flower position, for plant height, for pod color, seed texture. Look at this one, 6,022 yellow, 2,001 green. So he made these really important notes. And the cool thing was all of the ratios are about the same, 3-to-1. So something to note, a ratio is a total number, but then identifying what type you have of each. So 3-to-1 yellow to green means you have 3 yellow and 1 green, 4 total. So that's going to be 25% green, 75% yellow. It might look like it's saying 1/3 of it is green, but you have to total the numbers in a ratio. So 1 out of 4 is green. We're going to identify what he was talking about using a Punnett square. So a Punnett square is a theoretical breeding between organisms. Here I'm going to have a female, here I'm going to have a male. These are seeds. They represent what will become plants. And in these seeds, I can see some notation. I have a capital Y and I have a lowercase y. If it is capital, it's going to be dominant. And if it's lowercase, it's going to be recessive. So if this organism has both a capital and a lowercase, I know that this organism is heterozygous. Hetero being different, it has 2 different alleles. The same is going to be true for my male parent. It is a heterozygous plant. That means that when these plants produce sperm, they can put one of those alleles into one gamete and the other allele into the other gamete. They can pass on either allele that they have to their offspring. Now you can only pass on what you have. So heterozygotes can pass on both. Homozygotes can only pass on one type because they only have one type of allele. Something to note, it doesn't matter that the female's on top and the male's on the left. You can orient it any way you want. When we think about their theoretical gametes, we can then write them on top of a square that's divided into 4 and drop down those alleles to figure out what their theoretical offspring would be. So this theoretical offspring got both the dominant alleles from each of its parents. Here we have another theoretical offspring. It got the lowercase y from mom, and it got the uppercase y from dad. So this is a heterozygous baby. This was a homozygous dominant baby. Our next option is that you could get your capital Y from mom and your lowercase y from dad. So the outcome is the same, but where you got the allele is different. And sometimes that matters. Sometimes it changes the outcome if the dominant trait came from your parent that's female versus male. So we do want to consider both of them. That's why there's 2 of them, because who gave the dominant allele is different. And the last option is you could both give a recessive allele to your offspring. And so this is homozygous recessive. So I have 4 theoretical possibilities that could come from the breeding of 2 heterozygous parents. That outcome, if we look at the genotype, which is the number of alleles and types of alleles you have, is 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. That is 1 to 2 to 1, a total of 4. We could also do this in percentages. 50% homozygous dominant, 25% homozygous dominant, 25% homozygous recessive, 50% heterozygous. Then we can look at the phenotypic outcome. The phenotype is what you look like based on your alleles, but not what your alleles are. So your phenotype is that we have 75% that are yellow seeds and 25% that are green seeds. So we have 2 different ways to talk about our outcomes. Let's practice a Punnett square. So take a moment and try and complete this Punnett square, and then I will answer it. All right, so the purple flower is heterozygous. That means that the purple flower is going to have 1 big P and 1 little p. And a white flower, I can tell by looking at it that it's homozygous recessive. And I know that because it's showing the recessive phenotype. That means that it has to have 2 of the recessive allele. But I can also double check the question, and the question has shown me that it is indeed homozygous. So now I've got my genotypes of my parents up top. These are the theoretical alleles that they could put into their gametes. 50% of the gametes from this purple flower will have the capital allele. None of the gametes from the white flower will have a capital allele because it doesn't have a white flower to give. When I fill in my Punnett square, I just drop down those alleles. So I'm dropping down the capital allele from the purple flower, and now I'm dropping down the recessive allele from the purple flower. And the white flower will be able to donate a recessive allele to both of those. And my phenotypic outcome is 50% purple, 50% white. My genotypic outcome is 50% heterozygous, 50% homozygous recessive. You got to make sure that you say recessive or dominant when you say homozygous, because it could be either. Fabulous. Let's do another one. All right, now we have 2 purple flowers. One is heterozygous and one is homozygous. Take a moment to complete it yourself. Here is my heterozygote. Now, if I know the purple flower is homozygous, it must be homozygous dominant because it's expressing the dominant phenotype. So it's going to have 2 big P's Now when I fill in my Punnett square and I drop them down, based on the gametes that they could pass on to their offspring, I have 50% homozygous dominant and 50% heterozygous, 100% purple. Let's do it one more time. Now create a Punnett square for 2 heterozygous purple flowers. They're both heterozygous. I mean, go ahead and try it yourself first and then watch this. But they're both heterozygous, so they're both going to have 2 different types of alleles, one dominant, one recessive. When I fill in my Punnett square, one of the offspring will get both dominant alleles from the parents. 2 of the offspring will be heterozygous. One will get the dominant allele from mom, one will get the dominant allele from dad, and one of those offspring will be homozygous recessive. So I'll have 3 purple flowers, 1 white flower. I will have 2 heterozygotes, and I will have a homozygous dominant and a homozygous recessive. Cool. The last thing I want to show you for today's mini lecture is a meme. Heterozygotes, just allele uneven. And it's a picture of a goat that's half black and half white. And then it says allele uneven. I hope that you enjoy that. It's one of my favorite memes of all time. So Parker says hello. He's back here in the chair. And I hope you have a wonderful rest of your day. Email me anytime. Bye.